Yeah, so there's a couple other endpoints that I think are really important. The secondary endpoints that we looked at were that complete clearance endpoint, but at earlier treatment periods. So I mentioned that the primary endpoint was week 12. We also looked at complete clearance at weeks 9, 6, and 3. And all of those met the p-value of less than 0.05, with the exception of the week 3 endpoint in the Camp 2 trial. In Camp 2, we were numerically better than placebo for complete clearance, but it had a p-value that wasn't less than 0.05. So what that showed to me is that even early on in treatment, patients are getting a clinical response that they can see, and they're showing that they're going to see a difference in their molluscum contagiosum even at these earlier time points. In both Camp 1 and Camp 2, we didn't have any patients on VP102 who had a serious adverse event. The patients who had adverse events were mostly application site reactions, and this is related to the mechanism of action of the main ingredient in VP102. So the main ingredient is cantharidin, which is a vesicant, which means that it causes a blister to form and that lifts up and actually removes the lesion from the skin. And so based on that, you would anticipate, and we had planned for patients to have local skin reactions that were appropriate for that mechanism of action. So things like blister formation, discoloration of the skin, a little bit of redness, and things like that.